Hey class, welcome to Math 361, Probability and Statistics in the Health Sciences. My name is Dr. Schwab-McCoy. Feel free to call me Dr. SM or Dr. McCoy. I know the full last name is kind of a mouthful. As you probably know by now, I am at a conference, and so we are not meeting on Thursday, January 11th. If you woke up bright and early on Thursday, I am very, very sorry. Please check course announcements. I will let you know if any further conference or otherwise changes have to be made in our schedule. Because I am out of town right now, I decided it would be a good idea for us to spend a little bit of time still talking about statistics anyway by taking a look at a short video lecture. And I also have some quick assignments for you. I promise, these are quick. So the first thing that I'd like you to do, other than watch the video, which you are already doing, so high five, well done, is to take a look at the Math 361 syllabus quiz. This is a short assignment. It's about 10 questions that will hit the highlights of the syllabus for the course. So just about everything that I would expect you to know course policy-wise. To take a look at the syllabus itself, you can just go ahead and click on the syllabus link in blue line at any time. You can download either a PDF copy of the syllabus or you can find the exact same information written out here. So whichever format you prefer, take a look at the syllabus and complete the syllabus quiz. If you keep scrolling down, you will also find a course summary here. So this will give you a list of all of the due dates that are in right now. There are a few. All the things that I think will be covered on any given day in our course. Of course, that is subject to change, but this is kind of a rough outline of what I expect to cover as the semester progresses. So make sure you take a look at the syllabus and complete that syllabus quiz before Tuesday's class. The other thing that I'd like you to do is complete a short background survey. So this will give me an idea of a couple things. It'll tell me a little bit about your statistical preparation, what you know about stats so far. It'll also tell me a little bit about your career goals and what you hope to get out of this class. There are no right answers for the background survey. All students will get 10 points just for completing it, which is pretty awesome. This will just, again, help me to plan as the rest of the semester goes, and you can kind of see what sorts of questions I might ask you about. So again, no right answers. Tell me about yourself and what you hope to get out of this class. So before we get started with the content, I want to talk a little bit about what's in the modules tab so you can get an idea of what you can look for or what you should be able to expect out of me. You will be able to find course resources here in the modules class. This includes one of the 2 recommended textbooks for this course. You don't necessarily have to have access to both of them, but you should have access to at least one textbook. One of them happens to be free and open source and right here on Blue Line. Each set of modules or each lecture is going to come with a limited set of lecture slides to go along with it. I like to give these to you guys ahead of time just so you have an idea of what we're covering if you maybe miss class, or if you are kind of a slow writer like I am, to give you a bit more time to catch up during class. So what you'll see in these are examples. You'll see vocabulary words. What you will not see are answers and concepts. So we'll be talking about definitions, answers, key concepts in class. But this will hopefully give you a good starting point for your note-taking this semester. Again, these are all going to be available on Blue Line. Totally your choice whether or not you'd like to use them. So we are going to start in Module 1 today, and we're going to start at the very beginning with statistics and samples. So let's go ahead and jump in. Let's jump in at the start of the slide. Sorry about that. So one thing I like to do at the start of every statistics class is define statistics in case you're not familiar with what we're going to be doing this semester. So statistics is the study of methods to describe and measure aspects of some population from any given sample. So given data that we've collected, how can we take that data and translate it back to some population? A few other definitions that I really like are given below. One of them is especially apt. Statistics is the art and science of learning from data. As a statistician, statistics is a science in that there are a set of prescribed procedures, there are a set of methods, which we'll learn about a lot in this class. It's also an art in that no matter what statistician is looking at the data, I guarantee there will probably be some disagreements about how to proceed, how to define things, how to collect data, how to analyze the data. A lot of this is subject to our professional judgment, and that's something that I hope to see all of you develop as the semester progresses. Another nice definition for statistics is the study of how to collect, analyze, and draw conclusions from data. And this is a big one. We're going to try to make conclusions based on a data set. Throughout the semester, we're going to make use of what's called the statistical investigation process to study a data set. So every statistical analysis should follow these 4 steps. Step 1 is identifying a question or a problem. Step 2 is collecting relevant data on the research question. This is all module 1 of our course. This is the first step that we are really going to be focusing on in 361. What comes next is step 3. This is analyzing the data. And this might be more like what you would think of a traditional statistics class being. We're going to talk about a lot of different procedures for analyzing data throughout the semester. All of those procedures, no matter what we do in step 3, are going to lead back to step 4, forming a conclusion based on the data that we have to answer the research question. We don't want to come up with the research question and then just kind of let it fall by the wayside. We want to actually answer. So that's our final step. So we'll start out with some vocabulary, everybody's favorite. So as statisticians, when we're collecting data about a research question, what we're really trying to do is answer some question about a population. So a population is the entire collection of individual units that a researcher is interested in. Everything that could possibly be studied falls into the population. Populations are big, populations are dynamic, populations change quite often. So we rarely will have the entire population's data available. What's far more likely is that we'll be in a situation where we have a sample instead. So it's much more common that we will have a smaller set of individuals that we are selecting or sampling from the entire population. That's what our data is going to come from. So our data is going to come from a smaller subset or sample of an entire population. Now, when we're collecting data, what we're really usually trying to do is estimate or measure something called a parameter. So a parameter is a numerical summary describing a population. This might be, for example, the average GPA of all Creighton students. So parameters describe populations. Because I don't have information from my entire population, what I'll do is collect my sample and then calculate a similar numerical summary from my sample. So maybe I have a sample of 100 Creighton students, and I'll calculate the GPA for those 100. That GPA calculation, that average GPA, is going to be my statistic. It measures my sample. So ideally, what we hope is that this sample statistic, the information that we're getting from our smaller subset of data, is reflective of the population parameter. They may not match exactly the population parameter and the sample statistic, but we hope that they're close, and we hope that they're close enough that we can make some reasonable statements about the population. So that's our overall goal. So let's take a look at an example. The Current Population Survey is an important survey conducted by the U.S. government. It's actually ongoing. You may never have heard of this before, but it's happening all around you. It actually studies about 60,000 households every month, so it's a pretty big survey. The CPS produces things that you have undoubtedly heard before. Most recently, monthly unemployment numbers were recently released. That's one small piece of information from the CPS. Supplemental questions, these are additional questions, are added all the time. These are used as sort of a test platform. So, target population of the CPS. The CPS is actually trying to study households in the United States. So we are trying to collect information or measure information about American households. We don't have information about all of them. To give you a sense of scale, there are about 320 million adults in the United States. That's a pretty big group. So instead, we sample about 60,000 households per month. That's our smaller subset that we hope gives us an idea about all households or all adults in the US. So in May 2017, the unemployment rate according to the CPS was 4.3%. This is not a parameter. This is a statistic. The reason this 4.3% is a statistic is because it's calculated from our sample. We hope that it's a good estimate of the population unemployment rate, but really the best information that we had came from a sample. And that's what makes the unemployment rate actually a statistic, not a parameter. So collecting data, if we're going to take a sample from a population, we want to take that sample from the population in a way that is effective, in a way that gives us the best possible information. So we've got some strategies here. Ideally, we want to minimize sampling error and have a sample that represents our population. So first, what's sampling error? Sampling error is the difference between an estimate, usually a sample statistic, and the population parameter that's being estimated due to just random chance. So think about the households that are being sampled for the CPS every month. We're going to have different households every month. There's some fluctuations no matter what. Just because of the random chance of who was selected, there's going to be some changes in that statistic. So what we'd like to do is choose that statistic, choose that estimate in a way that we minimize the error, that we get as close to the parameter as we can. This is a key point though here. We have to understand as statisticians that samples will not be a perfect representation of the underlying population. We're going to miss something in our population, in other words. We'll set up our sample so that we represent the population as best as we can, as closely as we can get. One of the ways that we try to do that is to minimize bias, and we'll set up our data analysis procedures in such a way that this bias is minimized. Bias is the systematic discrepancy between the estimates, in this case the sample statistics, that we would get if we could sample a population again and again, and the true population parameter. So bias is that systematic error between the parameter and a statistic. What we'll try to do is choose statistics that are what we call unbiased, that get as close to the population parameter as we can get. They have no systematic discrepancy. One of the ways that we accomplish that is by taking what's called a random sample. So in a random sample, each member of the population has an equal and independent chance of being selected. This is just like drawing names out of a hat. If we choose people completely at random, that's actually one way that we get to minimize the bias between the true population parameter and the sample statistic. There's a catch. Random samples are really, really great, but they're not always feasible. And this is happening for a variety of reasons. There are lots of different scenarios where we cannot actually take a random sample. So here's one example where we may not have a random sample. An article was published on National Public Radio, NPR's website in fall 2012, that discussed recent advances in genomic sequencing and offered an opt-in poll. So people who are reading the article had the chance to then answer this poll. The question that was asked was, would you have your genome sequenced if you could afford it? The online poll stayed open for 2 weeks, during which time over 6,627 to be exact, votes were received. Do you think that this sample could be considered random? I'm going to say that this is probably not a random sample, and there's a few reasons for that. First, this is what we'll call an opt-in poll. People had to volunteer to participate in this NPR poll. The people who volunteered to participate might be a bit more passionate about the topic. Usually they're ones who have a strong opinion either way. The source of this is actually another reason why this isn't random. Not everybody in the United States has National Public Radio. Not everybody has the internet. Not everybody reads National Public Radio. That means that not everyone had a chance of being selected. So this isn't a truly random sample. That being said, there's probably not a lot of ways that NPR could have done this study otherwise. Even though this isn't a random sample, this might be the best information that they were able to get, given the resources that they have. Let's look at another example. So in a study, researchers took electrophysiological measurements from the brains of 2 rhesus macaques, monkeys. They're pretty cute. 40 neurons were tested in each monkey. This yielded a total of 80 measurements. So let's talk through a couple questions. Identify the target population. These macaques were probably chosen to try to represent all rhesus macaques, or all monkeys. So our target population is probably going to be all rhesus macaques. That's who we're interested in studying. Second question, do the 80 neurons constitute a random sample? Probably not. If we think about where these monkeys would have come from, they're probably lab subjects. So they were available to the researchers. We have 80 neurons, but really 40 of those neurons came from monkey 1, 40 of those neurons came from monkey 2. So we really only have 2 subjects that we're studying here. So we don't have a random sample of 80 neurons. We have 80 neurons that happen to be chosen from 2 monkeys who were probably available for research at the time and therefore not selected randomly. So, 3, researchers decided to replicate this study on humans. They used subjects who replied to an online ad looking for medical research volunteers. Is this an online, or is this a random sample again? Probably not. People volunteered. In order to get a random sample of humans, we would have to have some way to randomly sample humans. I would need to have a list of all possible subjects, all members of my population, in this case, all human adults. I would have to choose randomly from that list. I would have to be able to get people that I chose to sample at random to participate. Working with human subjects, it's pretty difficult to sample randomly. But that is okay. We will talk about different modifications that we can use, different other ways to incorporate randomness into a study. So the next topic that I want to talk about are types of variables. This may seem trivial to talk about what types of variables that we're collecting. It's actually pretty important because the type of variable that we have is going to inform how we can analyze our data. So what is a variable? A variable is just a characteristic that is recorded for each subject in the sample. So anything that I measure on my subjects or my observations or my patients, that's a variable. Again, these variables are classified based on the type of information that they contain. So variables can be either categorical. These are variables that describe membership in some category or a group. So these are ones that take on categorical values. Some examples include survival status. If a patient is either alive or dead at the end of a clinical trial, that's a category. Sex chromosome genotype. There are a couple of different genotypes that are observed in humans. Most commonly, XX for females, XY for males. A few other genotypes that we can observe, XO, XXY, XYY. These are all different categories for the sex chromosomes. Method of disease transmission. So the disease vector through which a patient obtains a disease, that would be categorical. Primary language spoken by a patient, again, also categorical. The important thing here, categorical variables are qualitative. They're describing some quality, some characteristic of the study or of the subject that cannot be measured numerically. On the other hand, numerical variables can be measured numerically. So these are measurements of individuals that are quantitative. They take on numbers. The numbers have magnitude. There's a relationship between the numbers. So a classic example of a variable that takes numerical values but is not numerical is Social Security number. Social Security numbers are made up of 9 numerical digits. It doesn't make sense to do any mathematical operations on them. You wouldn't want to subtract one person's Social Security number from another, or add one person's Social Security number to another. So even though those are numerical values, they are not numerical variables. There's no magnitude with the numbers. Examples of variables that are in fact numerical: core body temperature, age at death, number of amino acids. These are all things where the numbers have meaning. Now there is a slight distinction. There are 2 types of numerical variables. They may be discrete. They may represent counts. We may be able to list possibilities. They may be continuous. For the purposes of data analysis, it doesn't make much difference whether or not numerical variables are discrete or continuous, but it can affect things in a few situations. So let's identify the following variables as numerical or categorical. And I'll go ahead and just highlight them so we know which of the 2 they are. So let's take a look at the first one, number of injuries sustained in a fall. That is a numerical measurement. Fraction of birds in a large sample infected with avian flu virus. Now this one's kind of tricky. The fraction is actually going to end up being our sample statistic. Variables change on individual observation level. So for an individual bird, there's 2 options. The bird is either infected or it's not infected. So whether or not a bird is infected with an avian flu virus is categorical. Now, if I wanted to take a sample of birds from Nebraska and compare it to a sample of birds from Kansas, Colorado, etc., if the fraction is actually my variable, then I have a numerical variable. So it depends on what our variable actually is. Is it the infection status, or are we comparing the fractions? The logarithm of the body mass, that is a numerical measurement. Admitting diagnosis, what patients are diagnosed with when they're admitted to hospital, that is categorical. Weights of babies born in a hospital, weight is numerical. Sex, that is categorical. Finally, range of motion of the elbow joint of students enrolled in Math 361. Take a moment to think about this one. So here's your elbow. Here's the range of motion moving. We might describe this in terms of degrees, which is a numerical measurement. So how we're measuring variables can help us determine as well whether they're numerical or categorical. Now, we'll classify variables as numerical or categorical, but we'll classify them according to the role that they play in our research question as well. Variables can be either response or explanatory. So our response variable, this is our primary variable of interest. This is what we want to know the most about. This is what we care about the most. An explanatory variable, then, is just a secondary variable. This is one that helps us explain or may cause some changes in the response variable. The explanatory variable explains the response. The response is really more of the why. This is what we're most interested in. It's why we're doing the study. Let's take a look at some examples. So suppose we want to know whether or not cancer cells can be selectively targeted by using antibodies recognizing a tumor-specific protein marker. Researchers grafted human cancer cells onto healthy human mice. Some mice were then injected with tumor-specific antibodies while others were not. So our target population, what we are most interested in, see if you can guess. We are interested in the healthy adult mice. We want to know what happens when we are grafting cancerous cells onto the mice. So we'll take a sample of healthy adult mice. That's our population. Variables that were recorded. We've got a few things. We're recording whether or not the mice were injected with the tumor-specific antibodies. Some were, some weren't. We are also recording the number of mice exhibiting lymph node metastasis, exhibiting cancer signs in each group. Number of mice in each group would be a numerical value. However, our variable is actually whether or not the mice are exhibiting lymph node metastases. The number is just our summary. So both of our variables in this study are actually categorical. For each mouse, we're recording whether or not they were injected with the tumor antibodies, yes, no, and whether or not they exhibited signs of cancer, yes, no. So we have two categorical variables. Now our response variable, what we're most interested in is probably this one, probably whether or not the mice are exhibiting signs of cancer. We think that whether or not they have been injected with the tumor-specific antibodies would help us explain that. So our explanatory variable is going to be the infection, whether or not they have been injected with the tumor antibodies. So there we go. So I also want to talk a little bit about types of studies, how we can collect data. Data in the health sciences is usually obtained through either an experimental study or an observational study. So in an experimental study, the researcher manipulates the subjects in some way, usually through randomly assigning experimental conditions. And we'll talk a lot in this class about how to set up experimental studies. Sometimes, though, for whatever reason, we can't do an experimental study, in which case we may choose an observational study instead. In an observational study, the researcher does not manipulate or change variables in the study, merely observes. So in an experimental study, the researcher manipulates the subjects in some way. Observational study, researcher does not manipulate. They're just watching to see what happens. Reasons why one study might be preferred over the other. Cost of collecting data is certainly one. One of these two types of studies is usually much cheaper than the other. Observational studies are cheaper. Why are observational studies cheaper? Well, it's easier to just watch things than it is to actually do things to subjects. Second reason, feasibility, practicality of collecting data. For whatever reasons, sometimes an experimental study might be too difficult to do. If we're thinking about a 10-year clinical trial, it is really hard to manipulate subjects in a study for 10 years continuously. We may observe over a long-term period. If we need additional control, we might choose an experimental study. So the more control we want, maybe it's external conditions, maybe it's how the subject is behaving, more control we want, the more important it is to experiment. Experimental studies, manipulating those subjects, give us control. Finally, ethical concerns are a big thing as well. If we are interested in studying something like, say, a sexually transmitted disease, which may be harmful to patients, it's probably unethical to give patients the disease. It may be more likely to observe what happens in patients. So let's take a look at a few more examples before we wrap today's video up. For each of the following studies, identify the explanatory variable and the response variable, determine whether or not the study is experimental or observational, and determine the target population. I'd like you to try these on your own for just a moment. So number 1, researchers randomly assigned diabetes patients to 2 groups. In the first group, patients received a new drug called tasboglutide, where patients in the second group received standard treatment without the new drug. Researchers compared the rate of insulin release in the 2 groups. So our explanatory variable, see if you can think about this. It's going to be whether or not they receive the drug tasboglutide. Response variable is the rate of insulin release, the rate of releasing that synthetic sugar within the body. This is an experimental study because we are manipulating or deciding whether or not they receive the drug. Our target population is going to be all diabetes patients. We're interested in studying diabetes patients. We are choosing our sample from diabetes patients, so diabetes patients are the target population. Let's take a look at another example. Researchers in psychology tested whether the frequency of illegal drug use differs between people suffering from schizophrenia and people not having the disease. They measured drug use in a group of schizophrenia patients and compared it with that in a similar sized group of randomly chosen people. So our explanatory variable is schizophrenia status, whether the patient has it or whether they do not. Our response variable, think about this one for a moment, is drug use, whether or not they're having high drug use, low drug use, however we are measuring drug use. That is the response variable. This is an observational study. Here's why. I can't manipulate that explanatory variable. I cannot assign people to have schizophrenia. I can't assign people not to have schizophrenia. So we're merely just observing what's happening. Our target population is going to be all people with or without schizophrenia. We are choosing from people with both. We are taking our sample from people that are suffering from the disease and people that are not suffering from the disease. One other way to think about this, there are actually 2 target populations. We can think about this as population 1 being schizophrenia patients, population 2 being non-schizophrenia patients. Our final goal is probably going to be to compare those 2 populations. So that's going to be it. That's going to wrap us up for today's video lecture. If there are any topics from today's slides that you are uncomfortable with or unsure of, take a look at your textbook, email me. As I mentioned, one textbook is up on Blue Line, completely free and open source. The other textbook, Analysis of Biological Data, should be available at the bookstore if you'd like to take a look at that one as well. You can expect a quiz on Tuesday after we get back over this content, so you should be able to apply the definitions that we've talked about in today's class, virtual class, of course. Other things that I'd like for you to be able to do is complete the background survey and the syllabus quiz. These are both due Monday, January 15th. One final word about things that are due online. Anything that is submitted through the Blue Line system will always be due at 11:59 p.m. This includes any homeworks, that may be submitted online. This includes any quizzes, any project activities. Blue Line can sometimes take a few minutes to process submissions, especially if they are large submissions. So make sure to leave yourself plenty of time. Once the Blue Line system rolls over to 12 a.m., the due date has passed. So again, make sure to leave yourself plenty of time to get through that Blue Line submission system. That's all I have for you today. Hope you are all having a wonderful Tuesday, Thursday, whenever you are watching this between now and the January 16th. Hope you're having a great day, and I look forward to meeting all of you. Take care.